Good evening. We have a fairly full house, and it's 7 o'clock, so I thought I would start. I'm Josh Ginsberg. I'm the president of the Cary Institute. I arrived here in September, so I can now no longer say I am the new president of the Cary Institute. It's a pleasure to be here tonight, as it has been for all of the Friday night of the Cary seminars I've been able to attend, which is all of the Fridays. I missed the one that we did on Wednesday because I just didn't get my calendar right. This series is a really wonderful opportunity for people from the area to come and learn about environmental science broadly written. The topics range widely, as the sort of series of talks that I'll tell you about in a second will show. So tonight, I'll start with tonight, and we have our own Emma Rosi-Marshall. Emma has been at the Cary Institute for 6 years, is that right? Before that, she was a professor, got tenure at the University of Loyola in Chicago, and then decamped and came here, for which we are extremely grateful. Emma studies many different aspects of water and water ecology and systems ecology. She is known around here for many different things, but we particularly like the work she does out in the old part of the greenhouse that still stands, where there are 20 artificial streams, each of which has the name of a river from somewhere around the world. There is a Hudson River. And in those streams, she's able to study the dynamics of pollutants at different concentrations and look at their impacts on both water ecology and the organisms in the water. She'll talk more about that, I'm sure. But in the last few months, Emma's travel has been sort of symbolic of the confusion that I have had since I arrived at the Cary Institute. I was at the Wildlife Conservation Society for 18 years before I came here. They have a global program in 47 countries, and I thought, great, I want to be able to see my staff every day. We'll be able to have lunch together. We'll talk about the work they're doing in the Hudson River Valley, and we do that, but not every day. So Emma, when I was interviewing, you were in Australia. And then Emma took a trip to, I want to get the word, what is it, Argentina? Chile, Brazil. And then came back, and then went to Berlin. And then came back, and then went to Kenya, where she's doing a project near and dear to my heart, looking at hippos. You want to talk about hippos? No. Okay, next time. But it's basically looking at the way hippos move nutrients from the terrestrial environment to the aquatic environment, the rivers, and then the impact that has on everything else in the rivers. It's a wonderful project, and being a wildlife biologist who's spent much of my life in Africa, I think it's a great way of looking at the ecosystems. So Emma has been here for 6 years. She is a distinguished and valued member of the organization. I'm looking forward to her talk. But before we get on to that, let me tell you about the talks that are following Emma. On March 27th, so about a month from now to the day, we have Kurt Fausch from Colorado State University giving a talk on For the Love of Rivers. Kurt is one of the world's experts on the ecology of river systems, and is a great speaker, and it should be a wonderful talk. Following that, Rosamund Naylor, who's the director of the Stanford Center on Food Security, will be talking about feeding the world. You know, the next 2 billion people are going to be an increasing challenge for us to feed, because not only do we have 7 billion already, but many of those 7 billion people would like to live a better life. And so over the next 20, 30 years, as the population continues to grow, feeding 9 billion people is going to be significantly harder, more than just 30% harder, but probably twice as hard as feeding the current population of the Earth. And so Professor Naylor is going to give, I think, a very interesting and dynamic speech about how we're going to achieve that. We have a wonderful talk by Robin Moore, who's a natural history photographer, called For the Love of Frogs. That's on May 1st. And then we go back to another talk by a Cary scientist, Rick Osfeld. We'll talk about the ecology of Lyme disease, or weapons of mouse destruction, I think is the subtitle of his talk. But he'll talk about the ecology and how understanding the dynamics of ticks and mice and deer and foxes and coyotes, potentially mountain lions, will help us continue to go out into nature and be safe in nature and not get too scared about Lyme disease. So it will be an interesting series this spring, and I hope you can come to all or most of them. The Cary Institute, for those of you who don't know, is the nation's premier independent ecosystems research institute. We study the integration of the way the world works. So some places people study rivers, some people study streams, some people study terrestrial environments, and here we're interested in how things move between and amongst all those and really drive the processes that support life on Earth, not just ours, but the other 8 million species as well. It's a remarkable place. It is a place that I'm very glad to have landed, and I hope you enjoy yourself this evening. You will notice, and I have to point this out, on the backs of some seats there are labels, and those labels are for people who are members of our Aldo Leopold Society. One of the great perks of the Aldo Leopold Society is you can come here at 2 minutes to 7 because you know you'll have a seat reserved. So do consider supporting us. We rely on contributions from the general public for things like this summer series. So thank you very much for coming. And,
Okay, thank you, Josh, for that nice introduction, and thank you all for coming out on this cold winter night. It's nice to see so many friendly faces and new faces as well. So again, thank you so much for coming to the Cary Institute. I'm going to talk about my work focused on rivers on drugs. And so this is a picture of the beautiful Hudson River, which I'm going to talk to you a little bit more about as we go through. But I first want to start by telling you that this work is only possible because I have a number of people that work with me and then funders. So I want to acknowledge those foundations who have funded my research, and then in particular I want to acknowledge my collaborators, my postdoc Sylvia Lee, and my graduate students, undergraduates, and even high school students that have been working in my lab, pictured here. So it's really important to acknowledge the people that have made this work possible. So thank you. All right, I'm going to start with a quote from Rachel Carson, who wrote a book in 1962, which really did change the world, called Silent Spring. I assume many of you are familiar with this. What Rachel Carson said in this quote, I'll read it to you. The chemicals to which life is asked to make its adjustment are no longer merely the calcium and silica and copper and all of the rest of the minerals washed out of the rocks and carried into the rivers to the sea. They are the synthetic creation of man's inventive mind, brewed in his laboratories, having no counterparts in nature. Couldn't have said it better myself. The world that we live in today is not just affected by the things that were here in the past, but now we have created a lot of things in laboratories, and then we put them out into the environment, and we look and see. We, as ecologists, as myself, I want to see what they're doing to the world, because the organisms that live in rivers and streams and lakes have to adapt to these. And the research I'm going to talk about today is definitely linked to this quote. So what I'm going to focus on today are pharmaceuticals and personal care products. This is we often call them PPCPs. It's kind of a mouthful, but why we talk about them together is that pharmaceuticals and personal care products are things that we use in our everyday lives. They're very many different compounds. It's all the things that you have in your medicine cabinet, in your laundry room, to do wash, all the fragrances, all of those things that we use in our everyday lives. We put them all together because they have similar sources into the environment. So some of the things I'm going to talk about today include things like antibiotics, antihistamines, antidepressants, painkillers, anticonvulsants, antimicrobials, which is a little different from an antibiotic, hormones, fragrances, insect repellents, sunscreens, all of these things, all of these compounds that I've listed, that's a lot of numbers right there, but I want you to remember that when you go to the doctor and you have a bacterial infection, they don't just give you one antibiotic, right? There's many different kinds of antibiotics that your doctor could prescribe to you, right? We don't just have penicillin, we have many different kinds. And every one of these categories includes many different compounds that we use in our everyday lives. Indeed, sorry, I didn't even finish my list. Sunscreen, detergents, illicit drugs, cosmetics, all of these things, other things that I haven't thought of or we haven't yet invented, are included in this. And so indeed there are about 1,500 pharmaceuticals in the United States. So these are 1,500 drugs that we take, that we get a prescription for. But then you think about all of those personal care products, all these other things over here, which are not registered pharmaceuticals. There's many, many, many thousands of compounds out there that we use in our everyday lives. And I'm going to tell you today how they get into the environment and what they might be doing. This is an $80 billion global industry, just the pharmaceutical part of it. Huge industry, lots of money being made by development and sales of pharmaceuticals, and so it's a big industry. And of course, it does a lot of good for humans, right? We get better because we take medicine. So I don't want to disparage the fact that pharmaceuticals make us better in some ways, but there are unknown ecological consequences of all of these things that we use in our everyday lives. Okay, so how do drugs get into rivers? That's the first question you should probably ask yourself. So this is a beautiful river called the Salmon River in Idaho. It's called the River of No Return. It's in a wilderness area. It's beautiful. There's hardly anyone there. This river probably doesn't have very many pharmaceuticals because it's one of the most wild and scenic rivers in the United States. Unfortunately, the river right here, the Hudson River, is not quite as wild and scenic. And the big difference between the Salmon River and the Hudson River in New York are us, right? So the people. We live in the basin of the Hudson River, and because people are in this watershed, we influence the ecosystem. So I'm going to kind of walk you through the different ways that drugs get into rivers in the next part of this talk. First, I want to talk about manufacturing. So manufacturing, first we have to make the drugs. So where do we make the drugs, how do we make the drugs, and how is that linked to the environment? So currently, we have many different ways of manufacturing drugs, but there can be waste from the manufacturing process, right? So obviously you don't want to have too much waste or else you're not going to make money selling your drugs, but there are waste coming from manufacturing facilities that can be a big source of pharmaceuticals into the environment. So in New York State, for example, streams that have pharmaceutical manufacturing facilities have much higher concentrations of pharmaceuticals than areas that don't have manufacturing facilities. So we do have pharmaceutical manufacturing in New York State. This is a study that was done by Pat Phillips et al. With the U.S. Geological Survey. Just to walk you through this graph, this is a number of river sites where they went and measured the pharmaceuticals in the water column. This is the concentration here. In rivers where there's no hospital, that's what the NH is, versus a hospital, you can see there's not a big difference of having a hospital upstream of a wastewater treatment plant and then down into a river. But if you have pharmaceutical manufacturing facilities, you have much higher concentrations of pharmaceuticals in the river. So these are pharmaceuticals in the river. This is a log scale here. So these are much higher concentrations when there's a manufacturing facility than feeding into a wastewater treatment plant and then getting into a river system. So that's definitely the case in New York. A lot of our drugs, though, are not made in New York State. A lot of drugs are made around the world in India. And I like to think of this as, we talk about our ecological footprint a lot, and thanks to Michael Pollan, we think about where our food comes from. I'd like you all to start thinking about where your drugs come from. Okay, so a lot of drugs come from India. And so your pharmaceutical footprint is far removed from here in New York State. So here's India. This is a city in India. It's called Hyderabad, and there are many, many pharmaceutical manufacturing facilities in this city. Here's this city here, a Google Earth image. Here's the river going through. You can see it because it's a line of green. In this city, because of the way that we have developed industry around the world, it's a concentrated area for pharmaceutical manufacturing. So in this city, there's about 90 manufacturing facilities. They release their waste into one wastewater treatment plant, which then releases into this river. The concentrations of ciprofloxacin, which is a powerful antibiotic, which is made by some of these manufacturing facilities, in the river, the concentration is 32 milligrams per liter. Now that's a hard number. You don't really know what that means, maybe, but 32 milligrams per liter is really high for pharmaceuticals. And the reason it's so high is because of all these wastes from the manufacturing facilities. But just to put it in perspective for you, a typical dose of ciprofloxacin. So if you went to the doctor and you were feeling sick and they said you need a prescription of Cipro, they would give you a dose of somewhere between 250 and 750, depending on your weight, of course, milligrams. So that means that in 1 liter of river water, that's 13% of a dose in 1 liter of river water. So that's incredibly high. This is definitely a hot spot around the world. This is a place where the pharmaceuticals are really high. Now, if you think about antibiotics being in a river at that high of a concentration, you start thinking about antibiotic resistance, right? And whether or not the bacteria that live in that river are going to develop antibiotic resistance. And this group has actually done some of that work and found that. This is work done by Larson et al. In Sweden. This is what the river actually looks like. So they have many problems besides just the Cipro concentrations, right? This river is not healthy looking, right? So there's a lot of problems with that river, but I just want to show it to you as and these drugs are manufactured in India and then sold back to Europe or United States. So this is part of our ecological footprint. Okay, so the next thing we want to talk about is when we use drugs. So first we manufacture them, then they get sold, we buy them, we go to the store, and we buy drugs and we use them, right? And so we take them into our bodies, and it's important to kind of think about how often we use drugs. So in the U.S., about 3.5 billion prescriptions are filled per year. It's a lot. That's on average about 11.9 per person per year, and that's prescription drugs. Now you need to think about all the drugs that you have, that we all have in our houses that are over-the-counter drugs, non-prescription drugs. And then we got to think about all those personal care products as well. So this is a lot of things that we are using in our lives. I want to tell you that drugs, the metabolism of drugs in human body is very different depending on the compound that we're talking about. Now it makes sense that if you took a drug and it went into your stomach and then your stomach basically broke it down into its component parts, it wouldn't do very well as a drug to treat your illness, right? So our bodies typically don't do a lot of metabolism of the drugs. Meaning, what that means is the drugs tend to come out in our urine and our feces. We're going to talk a lot about human waste in this talk, so just so you know, because it's a big part of what I think about. Believe it or not. So the metabolism varies by compound depending on which drug it is, and also can vary depending on individuals. Actually, people respond to drugs differently, as you all know from probably your own experience, and so it really can vary. The other thing I want to point out is that a lot of the things, the personal care products that we use, that we put on our skin, that we wash our hands with, that we just put down our drain because we're using them to do our dishwasher or clothes, those of course are not being metabolized at all in our bodies. They're meant to go right down the drain. But the point of this slide is that when you take drugs, you might think that the pill is gone, but it's important to realize that you're taking that, it's dissolving in your body, and it's coming out in your waste. So it is not just going away, it's just going down the toilet. And now we're going to talk about what happens to it next. So then it goes, in an ideal world, it goes to the wastewater treatment plant. So here's a wastewater treatment plant. This is the second largest wastewater treatment plant in the world. It's in Chicago. The sanitary canal is right here. This is what it looks like if you're in a canoe. It's not the nicest place for a canoe ride because it smells like sewage, because the river in the sanitary canal is 97% of the discharge is from the wastewater treatment plant. So it's a lot, most of the river is wastewater effluent, actually, in the system. But wastewater treatment plants, you have to understand, are not designed to remove pharmaceuticals. Wastewater treatment plants are great. We all should support our wastewater treatment plant. If you haven't been to the one in your town, in Millbrook, for example, it is doing a great ecological service to us. It is taking our waste and breaking it down so that we don't have raw sewage going into our rivers, which was what we did 100 years ago, and we figured out that it caused us to get sick. So we built wastewater treatment plants. They're great. They're doing a lot of great things, removing pathogens, but they're not designed to remove pharmaceuticals. Sometimes pharmaceuticals will get broken down in a wastewater treatment plant. Again, it depends on the compound. A lot of times, pharmaceuticals will enter the wastewater treatment plant and come out in the effluent, in this liquid waste part of the wastewater treatment plant. This slide didn't reproduce terribly well, I'm sorry about that, but this is the Hudson River basin, and these yellow dots are all the wastewater treatment plants. So actually all the dots, the triangles are not wastewater treatment plants, but all of these are the wastewater treatment plants. Again, they're doing important service to us. By having wastewater treatment plants, we don't have raw sewage going into our systems, but they're not designed to remove pharmaceuticals. So there are a lot of them because they deal with all the people that live in the Hudson River basin. Okay, now we're going to talk a little bit about, okay, we've got two components of waste that goes into the wastewater treatment plant. There's the liquid, and then there's the solid. Okay? Just let you figure out what I mean by all that, the solid part of the waste. These are called, it used to be called sewage sludge. It's not called sludge anymore. We've renamed it biosolids. So the biosolids are, they're the part of the waste in the wastewater treatment plant that doesn't come out, that doesn't get discharged into the river. It's all the solid material. And 50% of that in the U.S. is actually land applied. And this is an image from actually Appleton, Wisconsin, where they're land applying their biosolids. Okay, so what they've done in this situation is they've dewatered them and got rid of the pathogens. So they're not spreading pathogens around from that waste, but they might be spreading pharmaceuticals around. So biosolids, solids, liquids, when our waste and all those pharmaceuticals that I just told you are in our waste stream that come down from our toilets, from us using them, when they get into the wastewater treatment plant, some of them, depending on their compound structure, some of them are going to be in liquid phase, are going to come out as liquids. Some of them are going to be, quote unquote, removed by the wastewater treatment plant, but that means they've sorbed onto the biosolids. Okay? And so then, potentially, you're adding drugs into the ecosystem through the solid phase of our waste. This is a complicated graph, but I really want to show it to you. This is from a survey of pharmaceuticals in biosolids. This was a nationwide survey. They went and took biosolids from all these different places. I'll help you through here. These are all antibiotics. So they took biosolids and they measured the pharmaceuticals residues in the biosolids. All of these are red. These are antibiotics. This is concentration here. It's on the log scale. You can see a whole bunch of different antibiotics. These are some antihistamines. This is Prozac, the antidepressant. This is triglycerin. We'll talk about that a little bit later. The antibacterial, which is very commonly found in the solid part of waste because it sorbs onto organics. Here's caffeine, which some of you may have a cup of next to your seat. And here it is in our biosolids. So we are taking biosolids. So I told you 50% are land applied. The other are incinerated or something else is done with them. I'm not exactly sure. But 50% of them are land applied. And so this is a potential source of pharmaceuticals to the environment. And when you land apply those pharmaceuticals associated with the biosolids, they can desorb. So they're sorbed in the wastewater treatment plant, but they can desorb. And studies have shown that they can come off of those biosolids and then enter aquatic ecosystems that way. Okay, solid waste, let's talk about that. Here we generate a lot of this stuff in our lives, right? So I talked to you about the drugs, but of course we also generate a lot of trash in our lives. The EPA has two recommended ways of disposing of pharmaceuticals, and we'll talk about this later. The first one is properly taking them to a take-back program to be incinerated. If you cannot do that, they recommend that you put them in your waste, mixing them with cat litter or with coffee grounds or something so someone doesn't get them, like an animal or a child or something, get into the drugs, and then you send them to the landfill. And so basically we've got a situation here where somewhere between 1,300 to 8,000 tons of pharmaceuticals are getting put into landfills every year. That seems like a lot of pharmaceuticals. So, and although we like to think of our landfills as being, within an impermeable, matrix, and it's not going to leak. They're not going to leak, right? But sometimes they do leak, and when they do, it's been found that pharmaceuticals can actually be part of that leachate coming out of landfills. So that's another source to the environment. In this part of the world, a lot of us have septic tanks, right? You maybe are not plumbed to the wastewater treatment plant. Maybe your house has a septic tank. And so for those of you who don't know, you have a tank, which then, your waste goes in. There's a lot of liquid waste. The solid sits at the bottom. Every couple of years, you're supposed to have that pumped out because the solid stuff is just going to sit there. And then it flows through, and you have this leach field, and then it absorbs or gets purified, the effluent. And so this is another potential source of pharmaceuticals into the environment. This is a study where they went and looked They took samples before septic tanks and after septic tanks. This is 2 different sites. The gray bars are before and the white bars are after. And just to show you a couple things, for example, caffeine was coming out of the effluent. Triclosan, the antibacterial ingredient in soaps, were coming out of the effluent in lower concentration. Again, triclosan tends to be sorbs to that solid waste. But you can see that these are other sources of pharmaceuticals to the environment. I want to talk a little bit about failing infrastructure. So I told you in an ideal world, our waste goes to our wastewater treatment plant. We don't live in an ideal world, of course. We live in the real world. And in the real world, we have situations like this. This is a manhole cover in Baltimore that clearly had a failure because this is all sewage on the ground in the forest. No one really noticed it because it was in the middle of the woods in Baltimore, except for Stuart Pickett took a picture of it. And this is a river in Baltimore that also has problems with failing infrastructure. So, for example, we've got about 32 billion gallons of wastewater flowing through our underground pipes in the United States. There's about 700,000 miles of underground pipes. That is a lot of mileage of pipes underground to deal with. Many of those pipes were put in about 100, 150 years ago. Out of sight, out of mind. We put them in the ground and we forgot about them. Right? Like they're there until someone's basement floods filled with raw sewage. We don't worry about them. And so what ends up happening is they're leaking, they're old, and there's about 900 billion gallons of sewage released into rivers and streams because of leaking infrastructure every year. This is an EPA estimate. In fact, the American Society of Civil Engineers recently gave the U.S. Sewage infrastructure a D. So we are failing to maintain our sewage infrastructure. They give similar grades to bridges and roads and other things as well. We're not so good at, again, once we build something, we just expect it to stay good forever. But our sewage infrastructure is in real trouble right now. The EPA estimates it will cost about $300 billion to upgrade the sewage systems in the U.S. Congress has allocated $2 billion per year over the next 20 years or something. That's 14% of what is needed. So we're really not investing. We're putting this problem off. Again, it's when someone's basement fills with sewage and they get a call, that's when people gets dealt with. I also want to point out that, of course, around the world, there are not wastewater treatment plants everywhere. Not everyone has built the kind of infrastructure as in need of repair as ours is. We still have it. The UN estimates that only 10% of the world's wastewater is treated, so that's 90% is not treated. 40% of the global population lives in coastal areas, and 80 to 90% of the sewage in these coastal areas is discharged raw into the coastal zones. So that's a lot of sewage going in because that's where many people in the world live, and then a lot of that is raw, untreated sewage, and raw, untreated sewage will have pharmaceuticals in it. Just a side note, it's estimated that about 200 million farmers grow crops in human waste, and this does not mean biosolids. This is night soil. This is collected human waste that is then applied onto agricultural fields, and the WHO estimates that about 10% of the world relies on this as a food source. There's obviously problems with pathogens associated with doing this, but there's also the problem of pharmaceutical residues. We also use drugs in our agriculture, right? So this is a picture of a chicken farm, if you will, and we use antibiotics in growing our animals. And so I've talked a lot about human sewage. We need to focus on also the animals that we also raise in this country in particular. So in the U.S., something like over half of the antibiotics used in the U.S. in terms of mass is for livestock. So we use a lot of antibiotics for livestock. There's a lot of concern. There was a presidential report. There was a CDC report that recently came out drawing links between the antibiotic use and raising animals and antibiotic resistance. It's a growing concern. So people have talked about that, but why I want to remind you of this is that I told you that our wastewater goes to wastewater treatment plants. Wastewater, all the poop and urine that comes out of these chickens that have been given antibiotics, they don't metabolize antibiotics either. It's in their waste, and their waste is either land applied or put into a lagoon or whatever's happening to it. A lot of it can end up in getting into the environment, and this is a contribution of PVCPs to the environment. And just so that you know this, more than half of the hogs and chickens in the U.S. are raised in large facilities like this that really that use antibiotics like this. So a lot of the meat that we raise in this country is raised that way. Okay, so what drugs, as a result, are found in rivers? This is a recent review. Basically, this is in wastewater, so this is 73 compounds of varying concentrations. Here in treated wastewater, it's 92. In surface waters, 91 compounds they found. In groundwater, they found 33 compounds. Basically, if you look for drugs, you can find them. And that's just the way it is. They're part of the aquatic ecosystems nowadays. What I, as an ecologist, an aquatic ecologist, am trying to figure out is, what does this mean? So we have drugs, and they get into rivers. Now what? What does this mean to the ecology of the river? Like, for example, the Hudson River. So the Hudson River has these compounds. These compounds have been detected in the Hudson River. This is a list compiled by a graduate student that works with me. These are the list of the compounds that have been detected. All in low concentrations, nanogram per liter. I told you about milligrams per liter at that river in India. These are nanogram per liter concentrations. There are many different compounds. They're entering the Hudson River. We don't know what the consequences are. That's what we're trying to figure out in my lab. But this is how I see the world. So the Hudson River is beautiful, and that's how you see it. But this is how I see the river. It's much more complicated than just a body of water. There's algae and bacteria and leaves and aquatic insects and fish, and all these things are connected to the terrestrial zone because bugs emerge and spiders eat them and birds eat them and bats eat them, and then things fall back in. And it's a complicated, what we call a tangled web. Right? So it's all these interactions, these ecological interactions occurring in rivers and in the terrestrial zone. So how do we untangle all these different compounds affecting all these potential different interactions? So that's what I'm going to talk about for the rest of my talk. Some of the things that we've tried to untangle in terms of what the effects are. First, I want to tell you a little bit about algae in streams. So if you go into a stream ecosystem and you start walking across a stream, you're going to slip and fall because the rocks are slippery. Why are the rocks slippery? Algae and bacteria. And so this is what it might look like. Here you look down, if you put your face in the water, you might see something like this. These are all cool little aquatic insects eating the algae, actually. And so there's like some just kind of green slime on the rocks. And you might not think much of it. This is, in fact, a rock that we pulled out of Wappingers Creek. So here's a rock. It's kind of just got a bunch of junk on the front, top of it. You know, you're like, oh, whatever, that's brown, mucky stuff. But if you look closer, you see something quite amazing. So here are these little strands of filamentous algae, and let's look even closer. And here are these beautiful algal cells. You look even closer, and these are diatoms that live on this. These are from Wappingers Creek. Beautiful little diatoms. They are glass frustules, so the outside of their cell has got this glass structure, and they're amazing diversity of species. So on this little rock here that just looks like brown slime is all this beautiful diversity, and this is what it looks like with all of its organelles intact. So really beautiful algae that live on the rocks in the stream right down at Wappingers Creek. So we've been trying to figure out what the effects of drugs are on algae. So one of the things that we've been doing is using our artificial stream facility. We grow algae in those streams from Wappingers Creek, and then we expose them to drugs. So here's an example here where we had a control, and then we exposed them to diphenhydramine, which is the active ingredient in Benadryl. And so what you can see here is that there's a significant decline in what we call gross primary production. That's how active those algal biofilms are growing. How quickly are they growing? And what you can see is you see a significant decline in how much they're growing in this experiment. We've also found other pharmaceuticals that suppress algae, triclosan, which is an antimicrobial compound that I'll talk a little bit about later, diphenhydramine, stimulants affect algae, and antidepressants. So these are just some of the compounds we've been working on, looking at whether or not they suppress this primary production. The other thing that we're doing with these streams is we're looking at the composition of the algal cells. This is work that's being led by my postdoc Sylvia Lee, who knows how to look at all these little frustules and count the holes and everything and tell which species is which. And so she's looking at the composition and how do drugs affect the composition of algal cells and communities that live on the rocks in our artificial streams. And she's also looking at whether or not drugs affect the size of the algal cells. So this is work that's ongoing. The other thing we're looking at are there's really interesting species interactions at this microscopic scale. This is a filamentous green algae here, this long green strand, and growing on it are these little teeny diatoms, this Cancanias diatoms. They're growing all on top of that, and this is a really important species interaction. These are always found growing on filamentous greens, and we're looking to see whether or not drugs disrupt these important species interactions as well. Now let's talk a little bit about bacteria. Many people think that bacteria is just something that makes you sick. Actually, bacteria they make ecosystems function. They are the base of most of the processes that as ecosystem ecologists we measure. And so in the autumn, for example, a bunch of leaves will fall into a stream, and they're going to decompose. They're decomposing right now. And the reason they're decomposing is because there's fungi and bacteria growing all on those leaves and breaking down all that organic matter and then feeding the aquatic insects as well. These bacteria, this is a stream in Baltimore. You can see this is sort of fluffy looking, both algae and bacteria growing on the bottom of the stream here. They're really important to aquatic ecosystems, important to the functioning of aquatic ecosystems. So we've been doing a lot of work on the effects of pharmaceuticals on bacteria. As I told you, antibiotics are found in streams and antimicrobials are found in streams, so it would make sense that antibiotics might affect these organisms. This is a study that we did on triclosan. So triclosan, if you don't mind a little audience participation, how many people have heard of triclosan? Oh, yes, that's great. So I've been doing a survey, global survey of how many people raise their hand. And you guys definitely come out as in the top knowing about triclosan. Maybe some of you have heard me talk about it, but that's fine. I'm glad to hear that people are learning about this compound. It's very, very common. It's in about 50% of our soaps in the United States. It's an antimicrobial compound. So it affects bacteria, algae, and fungi. And it's very commonly found in aquatic ecosystems, in particular in the sediments of aquatic ecosystems. But we were trying to determine whether or not it had an effect on that bacteria. So what we did was we grew we took artificial streams, we grew a bunch of bacteria and algae in those streams for a number of weeks, and then we added the triclosan to the stream in the concentrations that we find in the environment. So it's a pretty simple experiment. We have a bunch of streams without triclosan, a bunch with triclosan, and then we see what happens. What we found very quickly, actually after 7 days, was we saw an increase in the resistance of the bacteria to triclosan. So we didn't see, even though it's an antibacterial compound, we didn't see that the bacteria just went down. There wasn't like less bacteria. In fact, the respirations, we measured how much activity the microbial community had. It was the same in the triclosan treated versus the non-treated stream. And that's because the bacteria became resistant very quickly to triclosan. This is very important. There are a couple things that bacteria can do that we can't do, at least naturally. They can do what's called lateral gene transfer, so they can actually spread genetic material across species. If it's on a plasmid, it can actually spread through a microbial community, a bacterial community very quickly. So something like the resistance to a compound can spread. It's like us getting like, oh, I like that trait in my cat. I think I'll get it. It doesn't work that way. We can't do that, right? But bacteria, that can actually happen in bacteria. It's called lateral gene transfer, and so that can occur. And I don't know if that's what was happening in this system, but we did see rapid increase in triclosan resistance. Now, we can't look at bacteria under a microscope and identify them like we can with algae. So what we do is we do this what's called next generation sequencing. So we take a sample of that bacteria and we sequence the DNA, and then from that and looking at a clone library, we can figure out who is there. And when we did that in this experiment, we found that triclosan altered the composition of the bacterial communities. Big differences in the bacteria that were there in the stream. So not only were they resistant, but there was differences in the composition of these communities. We wanted to also go out into the field and see whether or not we could see anything like this happening out in a river. So again, here's this wastewater treatment plant in Chicago. We went above the wastewater treatment plant and below the wastewater treatment plant, and then we went to a smaller river in the Des Plaines River above and below a wastewater treatment plant. And we went and we took sediment samples. We measured the trickle sand. Remember, I've told you a couple times that it sorbs to organics and in sediments. It's typically not in the water column. It goes into the sediments because it's a really hydrophobic compound. So we took samples of the sediment, and then we wanted to see if the bacteria were resistant to trickle sand. If there was trickle sand in the sediment, do we find trickle sand resistant bacteria? And indeed we do. So as sediment trickle sand concentrations go up, you can see you have an increase in trickle sand resistant bacteria in the stream ecosystem. So they respond. These additions of wastewater treatment effluent into the system actually can promote trickle sand resistant bacteria. I do want to point out, though, I always have to point out that these high numbers here are not below this big, huge wastewater treatment plant. In fact, the high concentrations of trickle sand in the sediments were above the wastewater treatment plant. And you think, well, why is that? Well, if you've been to Chicago, this is the Chicago Sanitary Canal. It doesn't actually have any source of water. It was dug, right? It's a canal. And so upstream of here is all combined sewer overflow effluent into the system. So I didn't mention a combined sewer overflow, what that is. But that's in many systems in the United States. We figured out, like, well, when it rains, we have a problem. Our stormwater is going to come rushing in, potentially to our wastewater treatment plants. We don't want to do that. So instead, when it rains, we'll mix our sewage and our rainwater together, and we'll just release it to the environment that way, and they'll be diluted by the rainwater. And that's the system that was built in Chicago. They have now fixed it by this deep tunnel thing, but there was combined sewer overflow. That's the major source of water in this upstream area was from combined sewer overflow. So actually, the concentrations below the wastewater treatment plant, because wastewater treatment plants can actually remove trickle sand because it sorbs to the biosolids, it was actually lower concentrations below. Okay, so now a little bit more about bacteria and the effects of drugs. This was a study where we looked at common drugs, and we wanted to see whether or not it affected the activity of bacteria. And so this is just one little slide here. These are really common drugs. Caffeine, cimetidine is the active ingredient, and Tagamet, I'll tell you a little bit about that a little bit later. Ciprofloxacin is an antibiotic. Diphenhydramine is an antihistamine, and you can see that these drugs suppressed it's called the respiration of the microbial communities. That's their activity. So we basically take the pulse of these microbial communities, and we can see that when we expose them to these drugs, we actually suppress their activity. We also did this next generation sequencing of these bacterial communities, and we found that diphenhydramine, the active ingredient in Benadryl, changed the species composition, significantly changed the composition of these bacterial communities. Caffeine suppressed the activity, but it actually didn't change which bacteria was there. This kind of reminds us that everything's not going to be the same for pharmaceuticals in general. It's going to depend on the compounds. And so we saw different effects depending on the compound. All right, I want to tell you a little bit about some work that I'm doing in Baltimore. This is part of the Baltimore Ecosystem Study. It's a long-term ecological research site that we at the Cary Institute work on. We've been working there for over 15 years. It's in the city of Baltimore. We have a gradient of urban streams, that we've been working on. So that we've got forested, unurbanized streams up here, and then sort of suburban streams. And this is a really highly urbanized section of the Gwynns Falls in Baltimore. The long-term research has taken advantage of this urban gradient as a way to study how urbanization can affect the aquatic ecosystems. And people have been doing work on this for a long time here at the Cary Institute, and so I thought this would be a great opportunity to go and study the effects of pharmaceuticals in the system. I want to point out, you may have heard there was a report actually on Marketplace this week on this failing sewage infrastructure in Baltimore, and this is actually these are the sites that they were talking about. I work in those same sites. The infrastructure is failing in Baltimore. What's quite interesting about Baltimore, they put their system in quite late, actually, and by then they had figured out that combined sewer overflows were kind of a bad idea. Like, it wasn't such a good idea to just release a whole bunch of sewage, so they didn't put combined sewer overflows in. They have a separate storm system. When it rains, it's a separate system from this sewage. So in Baltimore, there shouldn't be any sewage in the rivers. But in fact, there is, and that's because there's a leaking infrastructure in the system. So we find pharmaceuticals present in urban streams, and what we're also finding is antibiotic resistant in the sediments, resistant bacteria in the sediments of these urban streams. And one thing that we find to be very interesting is that the microbial communities in these really urban streams are diverse, abundant, active, and resistant to antibiotics. So they're functioning microbial communities. They're not dead. Their urban streams are not dead. They're just behaving differently than other systems. And this is some work that's ongoing. Now I want to talk to you a little bit about bugs. Let's get a little bit bigger and talk about a bug. If you go to Wappingers Creek, like these school children have done, and put some boots on, put a net in, and kick around, you do the river dance, we call it, kick around the rocks, you're going to find some really cool things in Wappingers Creek. Really amazing bugs that live in the streams here. So things like this beautiful stonefly, mayfly, this really cool caddisfly that catches particles from the water column, the dragonfly, another mayfly. This is a really awesome Bellis dematid that is male because all the eggs are laid, the female lays all the eggs on the male's back and then he carries them around and protects them until they hatch. This is what I like to think of as like the lion of the streams of Wappingers Creek. It's a big, huge megalopteron about this big, and you can see it's eating a stonefly. It's an amazing photograph. So really cool bugs that live in the aquatic ecosystems just right down the way. Really amazing diversity. And so we're trying to figure out what happens when you put bugs on drugs. And so again, we use these artificial streams. In this situation, in this experiment I'm going to tell you about, we used a freshwater shrimp. It's a Gammarus fasciatus is its species name. And it completes its life cycle in the stream. So it can go from adult, have eggs, and then all the way through, so you can have multiple generations in a stream ecosystem. We gave this drug, we gave them Cimetidine, which is the active ingredient, and Tegamet. It's a H2 histamine antagonist. It is used for acid reflux. So if you have an overabundance of secretion, acid secretion in your stomach, you can take this histamine. I know we think of antihistamines as what we take when we have an allergy attack, but actually this is another kind of antihistamine that we use for stomach acid reflux. It was approved in 1977, the first drug to reach more than $1 billion in sales. It's a generic drug now, and it is very, very commonly detected in streams. Invertebrates, this is a really interesting thing to remember when we talk about pharmaceuticals and their effects on aquatic organisms. We are not that different from those cool aquatic insects that I showed you, but we are different in some ways, right? We all, we share common ancestors way, way far back, right? But invertebrates use the compounds that we have in our bodies, but for very different things, right? So invertebrates actually use histamines as neurotransmitters. So the way they smell, the way they sense light, the way they taste their food is actually communicated within their body using histamines. And so, ergo, if you give them antihistamines, what will happen to them? Right? So this was, it's a pretty obvious hypothesis, right? So if we know that this is found in rivers, and we know these are in rivers, and we know they use histamines, if we give them an antihistamine, what will they do? Well, what they did in this experiment was, here, this is the control, this is no antihistamine, these are different concentrations of antihistamines, and this is the number of individuals of this critter here. So what you can see is in the control we had lots of individuals, and then as we increased the concentration, we had lower number of individuals. And this number, this one right here, this concentration, is the concentration that Cimetidine is commonly detected in U.S. surface waters. So you can see that it wasn't like all the bugs died when they were given this drug, but their populations did not do as well. And so it really changed the dynamics of this population. We have some more recent research done by a graduate student, Aaron Richmond, that works with me on antidepressants, so things, selective serotonin reuptake inhibitors, SSRIs such as Prozac and Celebrex. We've been looking at them and what happens to aquatic invertebrates when you expose them to that. Again, we took bugs from Wappingers Creek in this experiment, a whole bunch of different aquatic invertebrates, and we exposed them to Cimetidine. I'm going to go back a second, to antidepressants. We exposed the bugs to antidepressants. And then we looked to see whether or not the composition of the community, we had lots of different types of bugs in there, we exposed them to these antidepressants, and then we looked to see who was there at the end of the experiment. And indeed, we didn't actually change the community. It wasn't like, again, there weren't like a whole bunch of dead bugs floating up and things like that. The bugs were still living in there, but we did see effects on the bugs. What we saw was that the invertebrates, these caddisflies, which are here, pictured here, beautiful aquatic insect, they spin nets. They're just a really cool aquatic insect, and they actually grew better when we exposed them to SSRIs. So we had a significant increase in the size, of the mean size of the individuals of the caddisflies. And then these chironomids, which are not exactly the most charismatic of the aquatic fauna, they just look like little, I don't know, they're just little bugs. But when they are adults, they're a little more exciting. They look like mosquitoes, but they're not. They're just lookalikes of mosquitoes. But they actually, when we expose them to SSRIs, they actually increase their emergence. So they sped up their life cycle. They seem to have moved faster. So it seems that in this case, this antidepressant actually increases what's happening with the bugs. We haven't done an experiment to look at the total biomass over time. That's probably coming in the future, but we definitely see that something's going on with these antidepressants, but in a different direction than the antihistamines I showed you. Okay, so what do drugs do in rivers? This is a work in progress. This is going to keep me busy for a long time because there's all these different interactions that I just told you a few things that drugs do. But there are a whole bunch of things that are happening in an ecosystem. How things grow, how things behave, right? So like we haven't even scratched the surface of figuring out, if you give a fish antidepressant or something, how does it respond differently? How does it change its behavior? How does the structure of the community change? I told you a little bit about algae and bacteria, but that's a kind of a big question. One of the things we're interested in our lab is how do they move through a food web? So these arrows, might be an indication, you could imagine, well, okay, the algae have some kind of pharmaceutical, and then the bugs eat those algae. Do they then get the pharmaceuticals, and then do they emerge, and then the bats eat the pharmaceuticals? All that could be happening. We just haven't investigated it yet. So there's a lot more to be done. I also want to leave you with the idea that these rivers have a mix. I like to think of it as a cocktail of drugs. Lots of different drugs are found in these systems. So this is this river in Baltimore that I work on that was featured in that story the other day on Marketplace. This is called Woodin's Run. You can see, I wish I could project the smell of this, but it's the river doesn't look so gray. It's gray. It's always got bubbles, and it smells not very good. And it's because, but again, there's no wastewater treatment plant upstream. There's no combined sewer overflows. This is just leaking infrastructure in this neighborhood of Baltimore. And a number of different compounds. This is actually the list from the Hudson River, but we have studied this system, and we're finding a number of different compounds. And why that's important is that we're trying to understand. I told you a little bit about, for example, what fluoxetine does or what diphenhydramine does. But you have to understand that out in the environment, in the real world, all these things are there. And you know that if you take a whole bunch of different drugs, right? Your doctor, before they prescribe, they want to know what drugs are you taking now, so that if we give you something, there's not some weird interaction. Well, think about that. Your river is like that, right? So the rivers have all these different compounds, and like the bugs are being exposed to a whole bunch of different things. We don't really know what the consequences are. And so this is, again, this could keep aquatic ecologists busy for a long time because there's lots of different things, and then trying to understand the interactions of these multiple stresses is a challenge. Again, I told you there's 1,467 pharmaceuticals in the U.S. and probably more, sometime soon. Plus, we have other stressors, right? So if you've been coming to talks at the Cary Institute, we have invasive species problems, and we have, native species declines, and we have climate change, and we have increases in nutrients, and we have a whole bunch of other things going on in our aquatic ecosystems affecting them. Drugs are sort of part of this picture, and trying to understand the consequences of all these things and how they interact to affect the health of our rivers and streams is really quite a challenge for us. You might be asking yourself, well, aren't these things regulated? Well, they are regulated sort of. The EPA regulates the release of sewage, but not pharmaceuticals specifically. They're not listed as aquatic contaminants under the Clean Water Act. They're just not. They're considered emerging contaminants or contaminants of emerging concern. We only just realized that they're in the water. Now it's going to take a lot before we actually regulate them. The FDA is who regulates drugs for safety, but they don't include, of course the FDA doesn't include the environment when they're regulating whether or not a drug, I mean they're trying to figure out whether or not it's safe for you to take. And then, so I do want to point out that the release for manufacturing facilities is starting to receive attention in New York State. I haven't kept up on this, but I have colleagues who've told me that they're, working with manufacturing facilities to try and, if you can stop it before it goes into the wastewater treatment plant, that can be a big benefit. So the effects of pharmaceuticals is not well understood, and it's also not well regulated. So, but I don't want to just leave you with, a bad feeling at the end of this talk that it's, all bad. You can do something. So I'm going to tell you what you can do after you leave here tonight. You can reduce your use of pharmaceuticals and personal care products. I don't mean the things that are, keeping you healthy. We all have to take drugs if we have an infection or if there's something wrong with us, we have to take drugs. But there are a lot of personal care products that we use that maybe we could use a little less of, or maybe we could decide that, the antibacterials, soap, soap works pretty well just on its own, not use certain compounds. So we could reduce our use of these compounds. It's very important to properly dispose of your unwanted medications. There are take-back programs. In fact, here they are in Dutchess County. So if you brought drugs with you tonight in hopes that you could get rid of them, you can take them to the Troop K headquarters. They're open 24 hours. I called them yesterday. So you can dispose of them there. They properly dispose of them. They incinerate them, which is the proper way to dispose of pharmaceuticals. But I put down a number of them for you. If you're interested, if these are places you live, but if you go to this website and you don't live in any of these places, if you live in Albany or something, there are take-back programs all over the place. So the DEC is really doing a great job taking back drugs. If you can't do a take-back program, then the EPA recommends disposing them as solid waste, but I would recommend take-back programs. You can also urge your local lawmakers, local, state, national, to maintain and upgrade our sewage infrastructure. That is really important. We need to invest more in this sewage infrastructure and in our wastewater treatment plants. They do a great service for us, but we tend to forget about them. Once they're built and doing something, we don't think about them until there's a big problem. So with that, I'm happy to take any questions, and thank you for your attention.
By what you're saying, I would imagine that water coming out of a headwater or right out of the hillside would have no pharmaceuticals, or does it go through the whole?
Oh, does it go through that lane? And yes, right. Headwater streams would not. If there's not a source of them, it's not going to come down in rain. As far as I know, although that could be an area of research, I have not done that. But I know there's some work where, like by a wastewater treatment plant or by a river that has lots of them, they can actually volatilize and from spray a little bit and get into it. But that's like particles, water particles. So yeah, if you're way high up in a headwater, you're not going to have much pharmaceuticals. Yeah, go ahead.
Would they even know how to get them out of the sewage?
Yeah, so there's really great research, especially in Europe. They've been doing a lot of work on the technology to get rid of pharmaceuticals in wastewater treatment plants. The thing is, it costs money. Like everything, it costs money. And right now, as I told you, we just have to at least make sure we get our sewage to our wastewater treatment plants. And then we could maybe think about investing in our wastewater treatment plants as well. But yeah, there are technologies available, not to get rid of everything, but they're working on it. Engineers are certainly in the fray and trying to make it so they're not coming out. Yeah, do you have a question?
Yeah, you were saying that individually the amounts of those drugs and stuff in the water was not a lot. Will you add them all together? Do you know what that amount is?
Well, yeah, so some people do that, like they kind of talk about the total pharmaceutical load. It's kind of, it's still going to be maybe in the nanogram, maybe in the low microgram per liter concentration. So low concentrations. But as you know, in order for a drug to have an effect, you don't need a lot of it to have an effect. And of course, an aquatic organism that lives 100% of its time in a low concentration, chronically exposed, it may have consequences for those organisms. And then especially when you add up all the different things that potentially are there. Good point. Another question? Yeah.
I get my drinking water from the Hudson.
Yeah.
The Poughkeepsie water plant. Am I getting?
I always get that question, so I'm putting this on. So this is not from the Hudson River, but This is finished drinking water, a nationwide survey. Again, this was a nationwide survey that was done here. And you can see that at high concentrations, this is 1,000 nanograms per liter. So these are low concentrations. You can see that in finished drinking water, you have fewer drugs at high concentrations and fewer drugs in general in drinking water. So they did include that. Now, the New York, I think it's the Department of Health, did a survey and measured pharmaceuticals in drinking water throughout the state. If you're interested, I can provide you with that report. I can't remember if the Hudson River right above the drinking water in Poughkeepsie was part of that. But typically the fact of the matter is what's in drinking water tends to be low because we regulate, we clean it up before it goes through our tap. So it's very low concentrations. And it's important to remember that, like, you think about your daily life and all the things we expose ourselves to kind of unknowingly, and then what comes out of the drinking water is probably going to be very low relative to all the other things in our lives. But certainly it's worth investigating, and that's why people have been measuring it in drinking water as well.
How about well water?
Well water, depending on, if you are too close to a septic system, there's, like another system, but most of the time it's going to be very low concentrations. But Russell Urban Mead, I believe, has done some work. He's local here and has done some work. He's with the Chasen Companies, has done some work on various trace elements in well water around New York State. So that's something you could investigate as well. So people are working on that. Yeah. Yeah, up in the back. How about?
Has there been any research done on reed beds in rural areas? There's enough room to use reed beds instead of sewage treatment plants. And do the plants there metabolize any of these compounds?
Yeah, so I'll repeat that question. She's asking whether or not you can use like a wetland as a treatment to remove these compounds. And again, the thing about pharmaceuticals, my answer is often the same. It depends on the compound. It probably also would depend on the wetland situation. So from what I know from the literature, people have been doing lots of nice experiments to figure out whether or not you can use wetlands as a treatment. And it turns out that some of the compounds, microbes in a wetland environment will metabolize these compounds, but other compounds won't. So, I mean, it's dependent on the compound. Yeah. Yeah, here.
So when you're testing chemicals, how do you take into account the breakdown of that chemical of what compounds, for example, refrigeration or
Great question. Thank you so much. It's a great question, which I didn't raise, so I'm going to repeat it. She asked about what about the breakdown, when chemicals break down, and how do I take that into account? This is actually, I should have mentioned this in my talk, so thank you for highlighting this. This is a concern as well as that the breakdown products of these compounds. So we measure, typically when someone goes out and measures pharmaceuticals in the environment, they look for the parent, we call it the parent compound. But in fact, sometimes the breakdown products could be just as important, and sometimes they're actually worse than the parent compound. And so chemists are starting to expand their repertoire of not just looking at the parent compound, but what those compounds break down into, and then people are doing investigations of, which compound is actually causing an effect. Because once things get into the environment, they're not going to necessarily stay the same. They may change due to UV or microbial activity into something else. And so kind of the question is, what does that do? And so chemists are working on that, and as ecologists, we're trying to figure out, breakdown products should be included in this big long list of pharmaceuticals. So thank you for that question. How about in the back here? Yeah.
The map you showed of Chicago with the waste canal, do those waste canals flow into the Chicago River?
Yes.
And then down to the Mississippi River, or do they go to
Yes. Chicago River, Illinois, down to the Mississippi. Yes. Currently, yes. Yes, they are doing. I mean, it is a really big wastewater treatment plant. They're doing a lot, and now the deep tunnel with the combined sewer overflow goes in the deep tunnel. If you want to know about Chicago waste and sewage treatment, it goes in the deep tunnel, and it goes to. So Chicago has the first largest wastewater treatment plant in the world on the south side, and the second largest on the north side. They like to build them big in Chicago. So the waste now goes to a holding pond and then goes into another wastewater treatment plant, because it is, of course, the drinking water for downstream Illinois counties, municipalities, and they're quite concerned about, how much is coming out in that system. So, yeah.
If we burn, some of our food piles, coal or whatever, should we burn our own drugs or?
probably not. It's a good question. No one's ever asked me that before. I don't know. You think in order to have an incinerator that would actually break down the compounds, you probably need to be really high temperature. So I probably would not. I would just take it to the state police office myself. But you could try it. I have no idea what would. And you would not want to do that because you don't know what it would. Might go into the air. So I don't recommend it.
Right. What are the products that break down better than others?
Oh, that's a really great question. So in Sweden, where they've really gone they're a little ahead of us in Sweden, they've actually tried to develop a database of pharmaceuticals that break down, that biodegrade much more readily than others. And you can actually, when you go to the doctor, you can ask for, can you check and see, is this one that biodegrades readily or not? And so, we can also think about, developing that.
The organic ones or the,
Yeah, I don't really have a good answer. I don't really know the breakdown rates of different things in terms of home products, but things like vinegar and baking soda are pretty benign, so those are a good place to start. Lemon juice, things like that are good. Yeah. Are people okay with staying? Maybe one more question in the back. How about in the back? One more.
Yeah. I use a zero water filter to remove the contaminants from my drinking water, which is city water from the Hudson. But there's still a great number of contaminants in it, presumably safe. I just don't want to consume them. But when I get rid of that filter, it's loaded with contaminants. And I did break it open once last year, and I put it in a flower pot, and the plants died.
Experiment.
So I was wondering if for the water treatment plants in the future, you're talking about Europe and how they're treating the filter system for a water treatment plant, how will they get rid of those filters? And is a septic system more effective because it goes through the ground, rocks and dirt, and it filters it out naturally? Of course, cities can't have something like that, but
Right.
Is a septic system a good one, better, more
I
effective?
can't answer the question of whether septic systems are better or not, because the research on septic systems is only just starting. And again, similar to wastewater treatment plants, it depends on the technology. It probably also depends on the soil, the percolation conditions, all these different things. There's a lot of things that are going to influence that. In terms of what to do with your filter, I would definitely recommend, whatever company sold it to you, see if they will take it back and properly dispose of it. That's what I would recommend. Same with where you buy drugs. Ask your pharmacist to take them back if you don't use them. But that would be my recommendation. But I think the boss is standing up.
So can we thank Emma again for an amazing talk. One of the other things we do is we have summer camp here, and because I have a 9-year-old, I've been focusing on the theme of the summer camp, and it's streams and stream health and ecology of streams. And Alan, please don't run that experiment where you break open water filters and kill everything. We don't know what's in there. But that's the kind of question that is just marvelous, because I think it opens up a whole other area of inquiry. I'd like to thank you all for coming. I'd like to just mention 2 other things. We do a lot of education work at various levels, and on getting the date right, March 21st from 9 to midday, we're doing a science and management forum on invasive species, which is being sponsored in coalition with the Duchess Land Conservancy. This is open to the public, to concerned citizens, but we really like it when municipal officials and others who are involved with the management of these things come as well. So please let your friends and colleagues know. And that you can register for that at the website or call to do so. Jessica, is registration necessarily in advance, or can you register on site? Yeah, we prefer that so we know how many people are coming. I just want to emphasize our next Friday night is the 27th of March, and it's Kurt Fausch from Love of Rivers. Should be a great thing. Thank you again for coming, and thank you again to our Alpha Leopold Society members for